the most important developments are those which are able to provide those single cells in a format which brings the cost of molecular analysis of single cells down. There are applications which are cognitive, so we study single cells to understand the heterogeneity of cells, so to understand, for example, how tumors develop, how metastases develop, and so on. Then we have diagnostic applications to be able to identify subclones which determine the fate, for example, of a disease, of a cancer, or of a response to therapy. And diagnostic applications are bound to therapeutic application, because if we can diagnose this in a more, I would say, subtle way, then we can also treat in a more precise manner. These are tools at every level. The pre-analytic part of a collection of those cells, depending on the tissue, is important to be able to collect those cells in a certain manner. For example, our field is to study rare circulating cells, so our target is to avoid losing cells, then to have those cells in a format where we can amplify the molecules that we target, DNA, RNA, microRNA, protein, metabolites, in order to be able to analyze them and possibly at low cost. Maybe the future that we are looking for are ways to automatize the analysis of single cells. In our field is very much important to collect them, that we do. We have a system to collect those rare cells from blood, but then the ability to study those single cells without losing them, and so to bring those analyses to the diagnostic market. I think that this, in a high throughput manner and low cost manner, that's really the target that we hope to see closer in 2019.